So with this second part, we will really enter into what we did really with Diatom metabarcoding. And we propose you this schedule. And in fact, you will recognize the different steps that Clarisse explained you before. But this time it will be really the application to Diatoms. And so the first point will be about the barcode choice. Second point, how do we preserve our samples? And this preservation experimentation, Agnès will present you what has been done. A few words about the SEN standardization. Third point, DNA extraction method. And fourth, I will give you our experience about the choice of the different sequencing technologies. Sorry, it should be a plural here. Okay, so let's start with the barcode choice. So I will present you several times this workflow. We start with a sampling, then we make a DNA extraction, then a PCR amplification, then we make a sequencing, a high throughput sequencing, and each sequence, so which is a line here, will be compared to a reference library thanks to a bioinformatic program here. This will give us an inventory, and then in the end, we calculate an index to assess the quality of our river. And so, again, we will see how, so which barcode do we need to use to identify our diatoms? It's always the first step of the process. How do we identify diatoms, which barcode to use? And I list here again the selection criteria to choose the barcodes. So if you learned clearly to what Clarisse told you, the first criteria is universality. Do you remember what we call universality? Is there someone who can tell me what is universality of a barcode? Ah, Daniela. Do you want to explain? It's written, in fact.
Didn't expect that, but okay, I can say.
That's a workshop.
Yeah, of course, I agree with you. Thank you. That is because of 5 minutes. Okay. Yes, when we're going, we're talking about a single barcode. Actually, when we were doing extraction of DNA of single specimen, a single species, then is universality, actually. Yeah.
Yes. In fact, the universality really the objective. So we have a diversity of diatoms. So if you imagine the tree of life and the diversity that we can represent, the idea is that this barcode will target the entire diatom diversity and not to lose some specific clades. So this is really this universality that we look for. The second point, variability. Do you remember what is variability? Why do we need this criteria of variability? Milos, you want to answer? Oh, great. Great. These are the people of BioLab project.
Yes. We have to learn this. Very, very good. So if I understood well, in the genome, we have introns and extrons, or the parts that are conserved and those that are variable. So variability means that we have to choose the region which is a bit conserved, so that the species from the diatom class or the taxonomy group has that gene, but has to be variable enough to distinguish species inside diatoms.
Perfect.
Thank you.
Do you agree with this definition? Yeah, okay. So that's it. An efficient barcode able to identify diatom species with conserved regions flanking this variable region to set the primers. Thank you very much, Milos. Specificity. Who wants to answer for specificity? Not always in the first. It's written, but we just for you to practice a little bit. why do we mean by specificity? Who wants to answer? It's written. Okay, maybe you. I don't know your name.
Or some sequence that is specific for Diatom class and not other species or
Yes,
class.
exactly. And why do we do that? Do you have an idea why? Why do we need to be quite specific of Diatoms and not to target all
because we target the Diatoms, that's what we need.
Yes,
As simple
yes.
as that.
And it will be a waste of sequencing power if
Yeah.
but it's difficult to be universal enough to target all the diatoms and not to take the neighbors like a chrysophyte and so on. Okay, thank you very much. The length, do you remember we talked about the length of the barcode? Why do we have criteria of length? Who wants to answer? Oh, your neighbor.
Okay, so basically the question was about the length of sequence that we're using in our research. Okay, so depends on which genes are we actually using. We saw that we can use any genome in living organisms, including nuclear genome, mitochondrial genome, and ribosomal genome. So basically, all those genes have different length. And for example, if I remember well, COX-1 gene was the shortest one, right? So if we basically decide to use that gene in our research, it will. I forgot what was the question once again.
The length.
The length, yes.
So yeah, think about the sequencing technologies. It's not possible to sequence the full genome. We have some restrictions.
Yeah. So basically, the machine is the one who is making those restrictions.
Exactly. So we have this restriction because of the sequencers. We can't sequence 1,000 length of a base pair, so it's too long. So we have sometimes these restrictions, and usually it's you said 300 yes, 300, 400. But it depends on the sequencer. So depending on what the platform, the sequencing platform offer you, you have to define the right length. Yeah, sorry, Agnès.
Want to add something on this lens because of course here we speak about Diatom, so we have good quality DNA, but sometimes we also want short barcode because the DNA we are looking for is a bit degraded, so will for sure not be able to amplify large sequences. For example, if we look at DNA that is free in the water, then sometimes it's better to have smaller barcodes that will for sure target the species we are looking for.
Yes, exactly. Thank you. And references. So who wants to answer? Let's continue the list. Let's stay in the same area, maybe in front of you. Sorry about that. Do you want to answer to this question? We were talking about reference criteria. Oh, okay.
So we need the references, so shortly to identify the species, because we actually, if we don't connect the sequence with some species, we don't have any results.
Exactly. Okay, we need some references. If not, it's not working. Okay, so we have our criteria. So now let's go to the supermarket or the market, and let's have a look to what we can find. And that was, in fact, the work of Lenaik Karmarek, or the first PhD thesis we had. So you see, 10 years ago. And so we look at the candidate markers that we could have based on this universality. So a single barcode that targets the entire diatom diversity. And we had this list: 18S, 28S, ITS, RBCL, and COX-1. And based on universality, variability, and reference criteria, we're going to see each of these criteria for each marker. We selected barcodes, and then we made a first in vitro test. She made the first in vitro test to really have a first selection. So let's first have a look to these two first points. Let's have a look to 18S. So you know what is 18S now. 18S, the good point is that references are available. There is a huge and good reference database. I told you that there was PR2, Silva, and really the first phylogenetic studies which were carried out by Linda Medlin in '93. So you see it's now quite old, but 30 years. You can see here in one of her first paper, the phylogeny. It was established based on 18S, so on this marker. So since 30 years, the number of references for Diatom improved year after year. So good point. Second point, we have several highly variable regions flanked by conserved regions. So what you told us, Milos, good point. And I show you here. This is an alignment. So maybe you can't see here, but here you have species name. For instance, here you have Chaetoceros diptebilis. And just in front of this name, you have its sequence, a part of its sequence, not the full sequence. And just below, you have another species, Coretron hysterix, the sequence here, and so on and so on. Here you have a Surirella. It's another species with its sequence. And you can see here, so we have a G in black, a T in red, a in green, and so on. All these sequences are aligned. To be comparable, we have to compare these sequences, we have to align them. So that's what we have. And you can see here, for instance, for this position, if you calculate this diversity index, this entropy index, it will be very low. Here also, if you go to this second column, do you see the colors here? This red color here, the second column, it's always the same nucleotide. So the diversity, the entropy index will be very low. But if you continue a little bit, here is a different story. We don't reach really to align them. So there is a diversity which is much higher. And so this is only on a small stretch. And here you can find here the diversity, the Shannon index, the entropy index along the different position from position 1 to almost to position 2000. And you can see here there are some very variable regions. And so yes, this region here that I focus on, it's this one here. It's the V4 region, which has been proposed as a barcode by Jonas Zimmerman in 2011. And it's a very good region because here you can put your primers that will be used for the PCR. So it's very variable. It means that from a species to another, you have a different sequence. So we are able to determine our species. So good point. Yes, 18S is the small subunit of eukaryotic ribosome. So it avoids the amplification of bacteria. So we were talking about a specificity, so it's also a good point. It's avoid one part of the tree of life, not all, but one part. Several primers already used for Sanger sequencing. So it means that some people already worked for us to define these primers. So also good points. We don't have to start from scratch. But some species can have identical 18S sequences. So not very good point, but okay, we see the list of good points. So it may be a good candidate for Diatoms. 28S, so it's longer than 18S, 3,300 base pair. 18S is 1,800 base pair. It's interesting for phylogenetic studies. It has been quite intensively used. There are several highly variable regions, and depending on the authors, different regions were sequenced. For instance, there is no homogeneity when you look at the papers along the years. There is no homogeneity. Some people use some particular highly variable regions like D1 and D2. Others use D1 and D3. Others at D1 and D4. So, and in fact, because of that, and also because of this length and the absence of generally accepted standard region, in fact, there were relatively few reference available for 28S. So this was a bad point here. ITS. So you remember, so this is the operon that showed you Clarisse. We have 18S, 28S, 5.8S. We have these ITS, which are the internal transcript spacers here and here. And they have very low selection pressure because they are XYZ after their transcription. So they are very, yes, they are very highly variable. So it's a good point, but they are not very adapted to phylogenetic studies. They are so highly variable that you have very big difficulties to align them from a species belonging to a genus to another genus. Here it's almost impossible. So which means that sometimes we have difficulties to compare these ITS. These ITS, they are more adapted to population genetics or for the biogeography of population inside species. So this is, yes, this highly variable version is a good point, but yes, and the other difficulty for ITS, sorry, is that for a single cell, you can have intragenomic variability. It means that inside a single cell, you can have different ITS sequences, which makes things difficult to be sequenced with Sanger sequencing. So the first generation of sequencers. And so, in fact, the number of references for ITS was quite low. References are available are poor. So 2 bad points. So not very interesting. So let's continue the market. RBCL, so just to remember you, it's a chloroplastic gene of 1,450 base pair. The good point is that it enabled to avoid all heterotrophic organisms. We only focus phototrophic. So this is a good point. It's a coding region. So it means that, so you can find it here again, an alignment. So we have our species name. Here, for instance, we have some Cyclothera species, Triata, Gamma, and so on. So you have your sequence and we align them. The good thing, since it's a coding region, we have no, like if I come back here to 18S, we don't have this kind of things. We don't have insertion or deletions. Where is it? Sorry, shouldn't do that. So it's easy to align. And since it's a coding region, we're able to translate into amino acid to check for stop codon. If you have a stop codon here, let's imagine we have a stop codon. In fact, it's a way to detect a sequencing mistake. And when filtering, it's an easy way to remove all these sequences, which are in fact sequencing errors. So good point. Good variability. By experience, we were able to distinguish cryptic diversity, so diversity inside species. So you can see here, again, this Shannon diversity index and the level of diversity of entropy along the position. So good point. And some primers are available. There are a few conserved regions inside the genes. You can see here. So maybe we can set our primers here. Also a good point because there are many references which are available because it's a marker which has been quite intensively used for phylogenetic studies. It's a good point too. Okay, let's continue the market with COX-1. So as you know, it's recognized at the standard barcode by the bold, so the barcoding of life, and Ebert and co-authors. It's a coding region, so this is a good point. Some references are available, but not so many, and you're going to understand why. Here you see the variability of the different position, and you almost don't find any conserved regions. And it's very difficult, in fact, to find universal primers inside diatoms. In fact, it's possible to find primers, but each primer should be specific of each genera. So it's really here it was a big problem with COX-1, and you will understand just after also. Yes, good variability. So you see the level of variability. So it enabled to distinguish cryptic diversity. Here, this is an example of a paper of Evans and Devinman. Maybe the diatomists know this is Selaphora pupula species complex, and each. So here, these are 6 cultures, and these 6 cultures, they were able to distinguish them with the COX-1. It was also possible with RBCL, but with less consistency. So just to tell you that COX-1, it's really powerful to go to subspecies level, but we have difficulties to find universal primers. So from this list, from these different criteria, we're selecting the following markers: 18S, RBCL, and COX-1. And what Lena Kermarek did, she compared their barcoding gaps. So just to remind you what is a barcoding gap, here you have a distance between 2 sequences on the horizontal axis, and the vertical axis is the number of distances, the percentage of distances. In red, you have intraspecific distances, and in yellow, you have interspecific distances. So a good case is when you have this barcoding gap, so when the intraspecific distances are smaller than the interspecific distances. And the bad case, is when you have this overlap. So what happens to our diatoms? So here you have 18S, RBCL, COX-1, and you have a number of distances with here the classes of distances. So here you have identical distances, and so when sequences are identical, and the more you go on the right, the more the sequences are different. What you can see here in green, we're going to start with the blue. Maybe you don't really see in the back. Here, you mostly have blue colors. It's intraspecific distances. When you compare species, when you compare sequences belonging to the same species. So low distances, of course. In red here, so it's mostly here for R18S, here for RBCL, and here for COX-1. In red, it's when you compare sequences belonging to two different species, but belonging to the same genus. So a little bit higher, but in many times, you see here for 18S, for RBCL, and for COX-1, we are in the bad case, so we have an overlap. And when you are in green, it's between sequences who belong to different genera. So it's higher distances. Just to make you understand that these intraspecific distances and these interspecific distances, you have an overlap in any case. Which means that there is no perfect barcoding, there is no marker with a perfect barcoding gap. So for this criteria, it's not working for any of a marker. So we continue our selection, and we made with this small set of barcodes, so 18S, RBCL, and COX-1, an in vitro test. So let's have a look to that. Is it okay with these first 2 points? Do you have questions or things which are not clear? No? Okay. So let's continue. What is an in vitro test? So you see again always the same workflow. We start with from a sample. We make a DNA extraction, PCR amplification, sequencing, and then we make an inventory. But here the idea was not to take an environmental sample, but to construct a sample of a known composition. And for this, what we did, we took some cultures which were already sequenced to know what we put in the mix. And we put in the mix the different cultures in known quantities. So these cultures, we put them together and we mixed together. And this test was carried out on 30 different cultures. And we made the test with 18S, COX-1, and RBCL. And this is the paper of Lena, one of her first paper, which was published in 2013. Here you have a list in fact, we mixed 21 different species. You have a list here, but we had 30 Diatom cultures. In fact, we wanted to see if it was possible to detect different species, but also to see if we were able to detect different subspecies. So this is why we have more cultures than species. For instance, for Gonphonema parvulum, we had some cultures which have slightly different barcodes, just to test if it was possible. So we mix the strains together. We extracted the DNA of the mix. We made a PCR amplification for COX-1, 18S, and RBCL. And then we made 454 Roche pyro sequencing. So Clarisse, she didn't talk about this sequencing methodology because it was a former next generation sequencing methodology. You're going to see that in fact it disappeared. So we sequence that, and all the sequences we compare them to DiatBarcode with two different algorithms. You didn't talk about this algorithm. It was 10 years ago because we are using new things now, which are more up to date. And then we compared the floristic lists with between COX-1, 18S, and RBCL. So the question is, which barcode is the most efficient to identify Diatom to species level? The first thing what we did, we looked at the proportion of reads matching with a single taxon. So what is a read? Do you know what is a read? Who wants to help me and define that? Sorry? Agnès. Okay. Okay. Oh, sorry. Yes, we need to introduce this term. You did this morning? So who remembers the definition of what is a read? Okay. Okay, Milos, okay, let's go.
I'm not sure, but when you are showing the sequencing picture, is this the read line which is connected with the name? With the you show, but okay, we don't have a name at the moment, but we have only this the how to say order of nucleotides, and it's one row in that.
So do we agree with that?
Yeah, I think that's the way the machine is reading the DNA of the amplicon. So it's the read is the sequence read by the machine, sequencing machine.
Yeah. And the sequence can be read several times. So we have a sequence. It can happen if a species is abundant in the sample, but this sequence is read several times. And when we have to analyze the data, we'll have to say, okay, I saw this sequence 1 time, 2 times, 3 times. We have to group together to say, okay, this sequence was found 1,000 times. So we also to simplify. So yes, we can have one sequence read 1,000 times. Okay.
Yeah, you explained that better now to me because okay, yes, that was one row. But if it's, for example, because it's from chloroplast, so DNA in chloroplast in Nichia species, Nichia, some species. So if she has 2 specimens and 4 chloroplasts and inside 10 DNA, so that means 40 reads will be in the sequencer, and we can see it.
Yeah.
That's
Yeah.
okay.
Yes, with all this heterogeneity of number of chloroplasts, number of genomes inside the chloroplast, and so on.
Yeah.
But we will see this after.
Plus the PCR amplification.
Yes, yeah. So just to come back to this graph, what you can see here, what we looked at is at different taxonomical levels. So you can see here we have subspecies level, species level, genus level, family, order, and subdivision. We looked at, for instance, for 18S, we see that 50% of the reads here are matching one species. So it means that 50%, are matching one subspecies. We are at the subspecies level. In the case of COX-1, all the reads match a single species. So it means, again, if we take the example of 18S, 50% of the reads could match 2 species or 3 or more. So first point, when you think about variability and so on, we talked about variability. The idea is to have one sequence different from another species. So here you can see that 18S is not so good. RBCL, for subspecies level, 90% of the reads match one subspecies. And then you see the evolution for species, genus, family, and so on. So you can see that when you are at the order level, most of the markers, let's say 90% or even 99% match only one taxon, so one order level. Okay, is it, do you understand? So COX-1 is the most efficient, so we are happy with COX-1. RBCL is okay, it's working, especially when we look at species level. And 18S has the lowest specificity. Which species are detected? So this is the reality. So the species we put in the mix with our cultures. So we know we have 21 species. With RBCL, we were able to detect 16 species and 5 were not detected. In fact, because they had low abundances in the mix, mostly the species with low abundances. With 18S, 21 species were detected, but 5 species were not detected. So this part of this red part, and 5 were added because simply we have this low variability. So we are matching sister species which are not corresponding exactly to what we put in the mix. And COX-1, the big problem of COX-1, in fact, 13 were not detected because the reference library was very poor and we don't reach to have a complete reference library. And then we can compare our floristic list. So you have here the reality. RBCL is the nearest, then we have 18S and then COX-1. So we are happy mostly with 18S and RBCL. So we selected, afterward you will see that we selected RBCL as barcode. And in fact, we refined the existing primers which were proposed by Stuf, Eichring in 2012 and Bruder and Medlin in 2007. In a paper of Valentin Vasselon, 2017, what we did, in fact, we took the reference database, so Diat barcode. So it was at that time the version 6. We made an alignment. So we had 638 species, and we selected a particular region. I think there was yes, the frame is not at the right position. Yeah, sorry, it's because of PowerPoint. But we selected a position of a stretch of 263 base pair, where we could put some primers and where there was this highly variable region. And so there was, you can see here, the forward primers, there are 3, this one, and this one, and 2 reverse primers. And maybe you don't see in the back, but they are degenerated nucleotide like this W here. So it means that we still have in this primer region some some variability in the alignment. So you can, if you want to do metabarcoding, we suggest to use these primers for RBCL. Okay, yes, you can find protocols. So if you really need to apply all that, we have protocols which are available for PCR. So in French, so there is this website. In fact, it's also presenting a Diatom reference library. But if you scroll down, you will find there are protocols, so they are in French. And if you are interested for English, you can go to this website. There is a DOI. It's on the website protocol.io. In fact, many people put their protocols there, and it's totally available. We can send also after the workshop all these links, so you can have them with you. Okay. Do you have questions about this first part about barcode choice? Oh, I'm looking at my watch.
I don't know this part. I know for 18S, we have 2 primers, 1 forward and 1 reverse. For RBCL, we have 3 forward and 2 reverse. A lot of people asking why is like that, and also asking, can you explain why this difference?
In fact, we have to come back again to this graph. We're probably going to be a little bit upset because I show you several times. Here, this is RBCL, and you can see the regions with low diversity. There is still some diversity. In fact, if you go to 18S, where is it? Here, we have really here some area, some stretch where the diversity is almost there is almost nothing. So it's possible to have single primers, not for RBCL. If we have these 3 forward primers with degenerated nucleotide, it's because we still have some diversity in these primers areas. Do you understand? Yeah.
Primer for RBCL was something like 27 bases long. So how does that function? Does the whole primer adhere to a sequence or some parts, and some nucleotides left to not bonded with DNA?
Again, I don't understand.
Well, primer is 27 base pair, not base pairs, but nucleotides long. So is that primer too specific, or how does that work?
In fact, they are quite specific. So if we come back here, they are not so specific. In fact, sorry. Yeah. Okay, that's not. Yeah. Okay. In fact, they are not so specific. I think we reach together all the diatom diversity, and even the neighbors, and the neighbors are stigmatophysi. Quite often in the runs, if we have low diatom biomass, and in fact it's catching some neighbors and phylogenetic neighbors, and stigmatophysi are catch with in our runs. I would say in the proportion, for instance, we had low biomass in high altitude lakes, and stigmatophysi. They don't represent a huge part of the reads, but let's say yes, 10%. So yeah, but it's always a threshold between all these criteria already presented you. Did I answer?
Yeah, pretty much.
Okay.
One question: do we use these primers in a single PCR, or do we use a separate PCR? Okay, once again, do you use a primer in a single PCR or in separate PCRs, and then mix the PCR
I need
products?
help here, please.
Yes, okay. So in fact, we use an equimolar proportion of these primers. reverse and forward, and we use them in a single PCR. Yes, and something also why we use why we tried to develop these 3 different forward and 2 reverse. It's because first we had this, we have more degenerescence in the primers, but then we saw that with this, they were too much degenerated, and it made us amplify. It was not specific enough, and we were also losing some diatoms. So that was for the aim of specificity and universality that we did that. It was working better like that. Because if you have too much degenerescence in one primer, then you may not catch very well the taxa you want, the target taxa, but you may also catch others that you don't want, and that's why we decided to do like that. From the reference database, we could see that it improved a lot.
Oh yeah, this is a really technical question, and in my head it looks like this. So the regions where on if you can show the graph with peaks. Okay, when we have this region, and left and right, where this is the let's say flat line, it's a conserved region. So which means A, T, G, and so on are the same for the most of the diatom species, and you have to put primers there because it's A, T, G, and so on. But sometimes between some species, it can be a change in one of these nucleotides, and you call it degeneration of primers. So you have to put 2 or 3 primers, which will slightly differ in order to catch and to bound the primer for all those species in the mix. That's why you need 3 primers and also. So I understood that well. Okay.
Exactly. Okay, maybe you can just show the primer sequences in the slide. Yeah, okay. Maybe just to show you here. For example, here you have G, G, A, and we could have degenerate that, but we did not because it was not efficient to degenerate. But here, still, we degenerate it with W. And here we put C and Y, which was partly degenerated. That's why it was a bit different.
Maybe you can keep the microphone.
Yeah, maybe. Can I keep this one? Is it a problem? Because it's not too long. I'm not going to be very long. Sorry. No, okay, I will take this one. Okay, perfect. Thank you. Okay. Can you hear me? Yes? Okay. So now I'm going to speak about sample preservation. Just in order, we change speakers so that we refresh a little bit. Ah, okay. So I have to change it. Okay. Now, did you open it? Do you see my share screen? Okay. Is that okay, Sergeant? Yeah, okay, perfect. So now I will speak about sample preservation. and about the Diatom sample preservation. Okay, so you know this workflow already per heart, and so we are going to speak about this first phase about sampling. Oh, because the beginning was yours, no? But it doesn't matter. Oh, yes, okay. So I will give the floor to myself. So, and there are several ways to preserve DNA. So probably you are used, those who are sampling Diatoms, you may have used formaldehyde to preserve your samples when you are going to do morphotaxonomy. But so the main thing that you have to know is that formaldehyde is not okay with DNA analysis. So don't use this preservation method, but you can use many other different preservation methods, which are ethanol, or use a buffer, or froze your samples. And we wanted to know if the different methods would have an impact on the DNA quality, quantity, and on the floristic list we could obtain from Diatom samples. So I give you that fine. Yes, it is. Okay, good. Okay, so now I'm going to tell you about an experiment we did at our lab. That was a collaboration with some colleagues in Europe, and the people that were in charge of this experiment were Cécile Chardon, who is leading the molecular biology lab in our lab, and Anna Baricevic, who is a colleague from Croatia, and we met in the DNAquanet network. So go again on the advantages of DNA metabarcoding, because it's, as we said numerous times, it's cost effective. One of the main advantages is that it's reproducible, it's comparable, and also you have these high throughput analysis. But some among the disadvantages comes that there is no standard protocols for now. And for example, different methods of sample preservation has been used up to now. And if you look at the bibliography, you will see that in different papers, you have different methods. And also, we had no clear information about the duration, how long you can keep your sample before you are going to sequence it. So the idea was really to compare different preservation conditions and also to compare different storage durations of the samples before the eDNA metabarcoding process. And the idea was really to bring scientific knowledge, but also to bring operational knowledge in order to go to further standardization later. We did that through the DNAquanet network with a workshop where we decided together how to proceed, and also through short-term scientific missions that were funded by this network. And in RAE, our lab has been leading the experiment and funding the sequencing. And we had participants from France, Croatia, Spain, Sweden, and Germany. So we decided to make the experiment on 6 contrasted European sites, 2 marine sites, and 4 river sites. So marine sites, you can see, I don't see, no, if you see my, no? Oh yes, here you have one in Spain and one in Croatia. And here you have a river in Spain, which is a mesotrophic river. We had an oligotrophic alpine river here by our lab. Here in Germany, we had a very eutrophic river. And finally, in Finland, we had a humic river. And in these sites, we, in the marine sites, so we took marine samples, of course, of phytoplankton. And for these samples, we made filtration of the water column sample and we preserved the samples as filters. For freshwater, we sampled benthic biofilms by scraping stones, following the standard, and we preserve the sample as pellet or as biofilm suspension, and I will show you that in more detail now. We selected from the bibliography 3 different preservation methods. One was cryopreservation, and for marine samples, we put it at -80, while for freshwater samples, we put it directly at -20. The second preservation method was to use ethanol and to keep the sample in a cool and dark place. And the last one was to use RNA later, which will limit the degradation of RNA, but you have some commercial buffers like that, but we use the homemade buffer, and we preserve them at -20 after the buffer was put. So with these 6 samples, we studied them at 6 successive dates through 1 year. So we take 1 sample of all these 6 sites after 1 day of preservation, after 1 week of preservation, after 1 month, 3 months, 6 months, and 12 months. Actually, we did also 1 year later, but we didn't analyze it. We just saw that it was coherent with the first findings. So all these samples that were collected at all these days were extracted. DNA was extracted with a commercial kit. And so you have 6 sites, you have 3 preservation methods. And at each site and preservation method, we made 2 DNA extraction to have replicates. So in total, we had 216 DNA samples. And we wanted to compare them for the quality of the DNA. So that's the famous ratio that Clarisse showed you today. And also for the quantity. So we measured the DNA concentration of the DNA. We also, for these samples, we proceed DNA metabarcoding with RBCL marker, and we made Illumina MySeq sequencing in order to obtain the floristic lists for each of these 216 samples. So let's first look at the DNA quality and quantity. On the top, you have the marine samples from Croatia and from Spain. And here you have the quantity of DNA. And you can see that, well, believe me, there is no significant differences between all them. But what we can see is that the concentration for marine samples is much smaller than the one from freshwater samples. But it's not at all the same samples, so it's not really an interesting point here, but you can see that. And also, I don't show you the results, but there was no observed impact on DNA quality. But what we can see, especially in the freshwater samples, is that the ethanol preservation, which is here in blue, here in yellow, you have the frozen concentration, the frozen condition, and here in gray, you have the RNA later, the buffer RNA later condition. You can see that at the beginning, well, all 3 methods seems okay, but then with ethanol, the concentration of DNA decreases throughout the time. Okay. What we looked also at the Diatom community assemblage. And here, for example, you have the number of OTUs. So the sequence that are clustered together according to a certain percentage of similarity, as Clarisse explained previously. So here the first two rows, you have the marine samples. And here, still in blue, you have the ethanol condition, the frozen condition, and the air annihilator condition. And you can see that, in fact, there is no real difference for marine samples, whatever the sampling preservation. Also here for freshwater samples, you can see that there is in each sample, there is no obvious differences between the diversity, whatever the preservation method. So although these sites still have higher richness, which is probably which is linked also to the higher DNA concentration. And you can see, and we did that for read numbers, for diversity values, and we observed the same for these three endpoints. And so we can see that the preservation methods have no impact on the read numbers, on the OTU richness, and on the diversity index values. If we go more in deep in the community composition on the diatom assemblages, here you could see still the same color code. And here you have the two marine sites. You see that all samples, the samples from the different methods and the different dates are quite grouped for the Spanish site and from the Croatian site. And you can see also that for each freshwater site, that was a very well clustered assemblages. So you can see that sampling sites have the main effect. And but when we go by site analysis, here, for example, for one of the freshwater sites on the oligotrophic, that's the site from France, you could see that the blue method is not exactly the same that the yellow one and the gray one. Okay, so there is some differences. But some differences between methods, but we could not, even with the site-by-site analysis, find differences between storage duration. So one of the most interesting questions was, are some taxa differentially detected? And in fact, when we look in more detail in these results, we could see that there is changes in relative abundances, but this is for abundant taxa. We have these changes that are modifying the repartition. And also we have changes in the presence absence, but only for low abundant data, taxa. And if we look at the overall number of taxa that are detected, that's over 300 taxa for all the samples, 81% of the taxa were detected by all the three preservation methods. Only below 5% of the taxa were detected only by one method. So that was also quite a good point. And the rare taxa were mostly method specific. That means that they usually appear or disappear without obvious pattern. Depending, we will find one on some date and another on another date. So it means that probably they are not very reliable. But when we go to the measure of ecological quality, and that is something that will come after in the presentation of Frédéric, so sorry for that, but maybe it will make more sense for you afterwards. So here you have the IPS score when for samples that are preserved with ethanol, with that are frozen or that are using RNA later. And you can see that whatever the site, the IPS scores that are based on the relative abundances of species based on read abundances, the IPS value are very stable. So whatever the preservation method, whatever the storage duration. So that's really a good point. because for biomonitoring purposes, probably you will not bring to the field a -80 device. And so that's right that for the moment, the DNA, the ethanol preservation is largely used, but also probably the preservation method with the RNA later will be also more and more used. So there is an overall robustness for biomonitoring purposes. But when it goes to more detailed analysis, then you may consider it differently because the ethanol preservation gave lower DNA concentration, but has no impact on the community composition and no impact on the IPS. And still ethanol is very rational method for field campaigns and storage, because then you can store them at cool temperature and dark, and that's the most easy way to do. So even in the worst case, when you had ethanol preservation, 1 year duration of the conservation, then you could see that the richness, the diversity, and IPS value were not affected. So that was really a good point in our sense. So, but when it goes to low density species, that was, for example, the cases for our colleagues with marine samples, phytoplankton samples, where they could be interested in looking at cyanobacteria that are going to bloom, or diatoms that are going to bloom and that are maybe toxic. And so if they want to have an early warning signal, that may be, you have to really think about the way to do it. And so now there's probably a need for operational standards. And Frédéric will speak about these standards later. And just if you are interested, there is this paper that comes from this experiment that has been released last year. Okay. You have some questions? Okay, Kalman. Je vais changer.
Thank you. Yeah, I'm just not sure if I understood it well. So the ethanol preservation showed that higher DNA concentration, right?
Sorry?
The ethanol method showed lower DNA
yes,
concentration,
yes.
but how did it? I mean, because when you add ethanol, you dilute the sample. So was it standardized to the original?
Yes, we try to standardize to have the same. So I didn't show you the full experimental design, but we standardized in order to have the same amount at the beginning for each sample.
Okay.
But what we think, it's probably that some cells in ethanol will open and free their DNA in the ethanol, and probably we lose this DNA because maybe you heard about the macroinvertebrates, people that try to conserve macroinvertebrates in ethanol and use the DNA in the ethanol to identify the macroinvertebrate assemblage. Mm-hmm. So we know that it's very easy for ethanol to take to catch the DNA molecules.
Okay, thanks.
So still about preservation. Quite early in the process, in fact, preservation of a sampling is fundamental. If you don't preserve it, you can't do anything. And so quite early with other colleagues working on DNA metabarcoding for diatoms, we realized that we needed to, in fact, to set some standards, some protocols. And we started with that, you see, in 2012 here, quite, so more than 10 years ago. And we proposed so first thing, to avoid formaldehyde. You understood it's not possible to use formaldehyde to make some DNA metabarcoding, but to propose in this protocol, in this standard, ethanol, RNA buffer, and deep freezing. And so we proposed this protocol to the European Committee for Standardization. It's called the CEN. in 2012. So I remember the first meeting we had. So all this group, you can see there is Alka Pulikova from Czech Republic, David Mann from UK, Martin Kelly from UK, Martin van Kuren from Croatia. I know that there are some Croatian here. Maria Keller from Sweden, Rosa Trobaro from Spain, Kunsab from Belgium, Jonas Zimmermann and Neil Anker from Germany, and ourselves. We proposed in 2012 this protocol. To be honest, it was the first time we were going there, and it was also the first time people were talking about DNA and to identify organism. And how do you say, the welcoming was not very warm. Okay, but we are registered, sorry. Okay, but so we proposed that in 2012. And so we went there several, in fact, every year we were proposing, and step by step, you see, 6 years after the publication of this technical report has been accepted at the European level. It was really a great job. We were really happy of that, because it's also a way to propose to everybody a single way to preserve a sample. So in fact, the way the sampling procedure is always the same. The strategy is the same. We take stones, we brush the stone, but the preservation products were proposed here in this technical report. Okay. So there are some protocols for sample preservation and also for sampling that are proposed here again on the same website in French and in English. So we will send you all these links if you're interested in for rivers and for lakes. In fact, we also propose some folders you can fill in the field if you're interested in. But of course, how you have to preserve and how you have to sample and preserve the diatoms. Or I proposed here a video, but I think it's not necessary. I think time is running, and okay, let's continue. And I think most of you know how to sample diatoms. Is it true? Yeah. So let's go now to the DNA extraction method. So we are here. So you see, progressively we move forward in the process. The fact is, for DNA extraction, so Clarisse showed you that there are different methods which are existing. And our question was, do this has an impact on DNA quantity, DNA quality, on the floristic list, and most important, on the diatom indices values when you make ecological assessment? If we use different extraction methods, does this has an impact on the assessment of the pollution level? So this was a work carried out by Valentin Vasselon. You can see here, it was published in 2017. And he carried out a test on 8 samples coming from several European countries, from lakes, rivers, and also from tropical areas. And we used 5 kits. I'm going to show you here these 5 kits. So each column here will be a kit. You have the 5 names here. And we're going to detail the different steps. And you will see that the strategies to extract the DNA is different. So here, this kit is called Mashrenagel kit soil. This is the soil kit, what we call also the soil kit. There is also from the same company a plant kit. Here, the S plant, it's another kit, a kit for blood. And here, this is the Genelute, the well-known methodology to extract DNA. Here, this is the first step for DNA extraction. It's when you explode the cells, the cellular lysis. So for the soil kit, it can be done with, it's mechanical, it's with some small beads. For plants, it's enzymatic with RNAs. With the plant kit, it's also enzymatic, but with proteinase K. And with blood, it's also with proteinase K. With Genelute, we do different things. There is a thermal shock, there is a sonication, and it's also enzymatic cellular lysis. So you can see here, different things are done. The second step is the lysate purification. So for these kits, these three first kits, the contaminants are retained with the filtration column that also you showed us before. The blood kit, there is no column filtration, and then for the genelute, yes, contaminants are removed during the centrifugation step. So here you see also we have differences. For DNA isolation, so it's made either by adsorption. So here it's with some colons. Here you can see some silicate colons. Here it's done with precipitation. Yes, with a buffer. And the last step, the final elution, it's here in the manufacturer buffer, and here it's done with resuspension into water. So does this has an impact on our different criteria? The first thing which we looked at is the quality and the DNA quantity and the DNA quality. And what we saw is that kits with high DNA quantities, which unable to have high DNA quantities, had low DNA qualities. In fact, we had some PCR inhibitors like this kit SAG. And kits with low DNA quantities have good DNA qualities, like soil kits and kits with silicate colon. And so there is really a balance between these two criteria. And really, it's important to prioritize quality in our case with diatoms, and to avoid to have all these PCR inhibitors. And this is by experience that we saw that there are many rivers or lakes where you have some humic acid, and this is not good for PCR. It's really problematic, and you lose these samples if you don't have this silicate colon. So yes, privilege the quality instead of the quantity. Usually we have no problem with diatom DNA quantity. Second question, after sequencing, is there an impact on community structure? So this is an MDS. Each color is a sample. So you can see that we have how many? 8 samples, 8 different colors, and each point is a different kit, and we have some replicates for each kit. So I guess, for instance, these are coming from the same kit, and there are 2 replicates. And what you can see, yes, so it's a different way to represent the results this time. You have it's a cluster analysis, and each bar is corresponding to a species. And you can see here that, for instance, this is one sample, and you can see that even if you're using different kits, the proportions in the species are respected, even if we have sometimes small differences. And what we could see is that there were no significant differences between the kits. And yes, the major significance is really between the samples. Again, always the same question. We have our floristic list. We make the formula. We transform this with the formula of Zelinka and Marvan to assess the quality. Here you have the IPS. So this is the index you're using in Serbia. So 0, it means it's a bad water quality, 20 high water quality. So when you are in the blue, it's a very good water quality. So what you can see here it's one sample, in blue it's microscopy, and here the 5 different kits. Again, we have no significant difference in terms of water quality when we compare the different kits. No impact on the index value. So this is the result of the paper, but our choice really is to use this soil from Mashrenagel, the nucleospin soil. Because yes, it's by experience we have good result with this, and we lose almost no sample. Again, the protocols are available in French. You can download them always on the same website. And here there also in English a DOI where you can find them. Do you have questions about these DNA extraction methods and how to proceed to select this kit? Oh, very calm. Yeah, good question. Yeah, yeah. Do you have experience with other kits? Yeah, Daniela.
Have more experience comparing saline habitat and freshwater with the same kit. It's a soil kit. With saline habitat, we had a problem, and probably we will have in May in your institute when we check our samples, that the quality of DNA was bad. Actually, I'm afraid to continue with PCR and everything, because in Sava and Tisa, like in freshwater, I didn't have this problem. I have good quality and good quantity, but in saline was very bad. And I repeat a couple times and the same bad. Then I decide, okay, I will go forward and try that. And because I didn't have options, I couldn't repeat the sampling. And actually, at the end, we had the good results. Also, Demetrio proposed me to wash samples from saline habitat a couple of times before I start DNA extraction. I did that, but actually I always had the quality was the bad. But at the end, we get sequencing and everything was fine. Then definitely we need to be more careful with saline sample.
Yes. So the quality was bad.
Yeah.
But the PCR were working.
Yeah, PCR working, electrophoresis, everything, and now I have sequencing and everything is fine.
Okay.
Then definitely the choice of the kit is good.
Yeah, but and the thing is probably that the kind of biofilm you use were really not included in what we experienced, where they were biofilm from rivers or biofilm from rivers, and here was it different? It was probably sediments, or
it was sediment, mud, because actually in all our sites, saline and freshwater, we have mud.
Okay.
Yes, so to use the knowledge that we get in the morning for the first time, I heard that this soil kit is based on silica column, and then low pH and high salts are important to bind DNA, and to release DNA high pH. Is it right? Okay, saline habitats that we are investigating have very high pH, and they're alkaline. So if we don't flush them, it means that we cannot achieve low pH, and DNA will not bind to the soil kit, to the matrix. So that's why probably Daniela succeeded to get any DNA after washing. So we have to take into account that. Does this kit works in marine habitats, soil kit?
Which kit was used for the preservation method?
It was this kit, but it was filters, so there was not a lot of salt probably remaining on the filter. So it was quite different from sediments.
And yes, because you have experience on marine habitat, Clarisse, what kind of extraction kits are you using?
I was using the phenol chloroform extraction protocol with which doesn't use this currents, so I didn't try with those.
Okay. Kalman has a question.
It's not really a question, but now I am also analyzing sediment samples, and actually in Hungary, this kit what you use, and I also use with you, it looks a bit more difficult to have it, and now I'm using the Quaigene's Power Soil kit, and I don't know if you have experience about it or somebody has, but maybe it can be also very it can be a powerful kit for such purpose.
Do you have good success with it?
I don't have results yet.
Okay.
I just started. I extracted DNA, but yeah, but I don't know.
Okay. And PCR? What with PCR? Not yet.
No.
Ah, it's really.
Yeah,
recent?
it's really,
Okay.
it's really no.
Okay. Other reactions? Okay, we can move on. Okay, so the last point is about the choice of the sequencing technology. So we are here, you see that we progress progressively. I hope you are happy of that. So yes, there are several points I would like to list here. The first one is that there is a really quick evolution of the sequencing technologies. And I have the impression that I think it's true, more and more different technologies were available on the market. And also inside each technology, there are some quick evolutions. And I would like also to add that some disappeared. So this is the first point. There is also a cost reduction, which is the second point. And the third point, there is a need to deal with the availabilities of the sequencing platforms. So in fact, to be honest, what was my impression when we started to have all this experience with DNA metabarcoding, when you speak to the sequencing platform, they propose many different technologies, and it's more like this forest, a tropical forest, where you have to push the branch, and this just to talk about our experience and what we did during these couple of years. So our first two publications with Lena Kermarek, our first PhD students, were carried out on Roche 454 pyrosequencing. These are the technical points that we had. The ring length, you can see it's quite long. We have 414 base pair. The error rate was 0.1%, so it's a very good, it's a very low error rate. Here you have the number of reads per runs, 1,000 and 100, around 100,000, which is quite low. But at that time we were very happy to have all these reads per run. And it gave good results in terms of sequencing quality, yes, with this low error rate. But it was quite expensive. I remember that we had 2 runs during the PhD of Lena, and yes, it was quite expensive at that time, and we were really happy because we had a small project but unable to finance this. Yes, and what is important to say is that 454, it arrived on the market in 2004, and they stopped, no more support after 2013. So it disappeared from the planet. So and just to tell you that sometimes you develop some methodologies, so you push a lot of effort in that. I mean, personally, our lab, and sometimes, in fact, the sequencing platform, the sequencing technology are not anymore supported. So you need to be a little bit aware of that sometimes. No? We are quite dependent on that. And we had also several papers with another technology, which is called PGM, ion torrent, which is not based. So this morning, you showed us, no, not yes, this afternoon, you showed us, Clarisse, what we read the sequences with this fluorescence nucleotide. Here it's not, I don't remember, I'm not going to enter into the details because it's quite technical and I don't remember exactly how it's working, but it's based on pH. It's detecting, depending on the nucleotide, is based on pH detection. And what to read? Yes, in terms of read length, yes, we target 263 base pair plus primers. It's 312 base pair. This is the number of reads we get, 6,000 per run. And the very good point, at least for us, it was quite cheap, much cheaper than Illumina and 450, because at that time, you see the years. So this is the papers. So you have to remove 3 or 4 years for the sequencing. So it was yes, 2014, 2012. At that time, Illumina was still quite expensive, and they reduced progressively. But at that time, Ion Torrent was a very good solution. The only problem was that the sequencing, the error rate was of 2%. And in fact, there were many problems with poly A. When you look at the alignment, here you can see, so you have your different species and the sequence. In the barcode, you have a stretch where you have, so it's 4, 5, 6, 7 A. It's what we call a poly A, a stretch. And Ion Torrent had big difficulties in sequencing these areas, and there were some, as you can see here, some deletions. In fact, you don't have deletions in reality. So it was really problems even in the dominant sequences that we had with Ion Torrent. But we published several papers, even if we had these problems here. Illumina, so with fluorescence, we have also several papers with this technology. Yes, there are different platforms which exist. ISEC, so Illumina is a kind of technology, and inside this technology, you have different possibilities, different versions. You can see the different names here. And I would like to give you an example with MySec and V2. V2 is a kind of chemistry that we use several times. And here you can see the read length is 2, so 1 forward and 1 reverse with 2050 base pair. But you can select different chemistries with 150 or 300 base pairs, depending on what you want to do. If you have some barcodes which are a little bit longer or shorter, you can select what you want. Here the error rate is yes, it's quite low, and I think it's even lower from our experience. 10 million reads per run, this about 6 to 7 gigabytes per run, and the cost that we have, at least on the platform of our institutes, is about 4,000. It's a little bit more expensive when you go on private platforms. And yes, really, if you want to work on endemism, that's what we did on our last paper on rare events. Really, you have a good robustness with this technology because of this low error rate. Another example, nanomysec. So when you see the different names here, for instance, Novasec is giving a very high amount of reads. I don't remember, but it's much higher than 10 millions. But with a smaller stretch, smaller, I think it's 150 base pair. If you want to make some small runs for trial to see if, for instance, your primers are working well, or if you have a smaller number of samples, you can use this nanomysec. You see here the number of gigabytes. It's much less, but it enabled to have some small tests. And what did I wrote? Illumina is widely deployed in the sequencing platforms. Yes, these are 2 platforms where we are sending our samples. There are many different versions, so you can find the good option for what you have to do. Okay. Another sequencing technology, nanopore. So maybe you already saw on this kind of very small sequencer. So this is the mini ion. There are others, let's say, I don't know how you can say platform or sequencer. Yes, it's a sequencer, and inside you have a small device which is called the fungal. Here you can parallelize this kind of small cell here. So you have small, this is the smallest, medium, and the biggest size. Here, the interest is that you can sequence long sequences of several tens of kilobytes. So we are really not in the same area that we have with Illumina. And usually the sequencers are used for genome reconstruction. Here, the problem for us, if we work on metabarcoding, the sequencing mistake error is of 5%. This is the amount of data you get with one thing like that, so mini ion. And we used with so Valentin Vasselon and comment s'appelle, Rémi Marcel, sorry. We use this mini ion plus this fungal, so you can see here its exact name. And in fact, the idea was to see if we sequence two different lengths. So you remember we talked about this 263 base pair because we had to adapt it to Illumina. So we wanted to compare if we sequence this small stretch with Illumina, but also this small stretch with this mini ion, and to use the capacity of mini ion to sequence the full length of RBCL, which is 1,473 base pair, so the full RBCL, and see what are the results for the identification. And here are the results. So this is one sample, a second sample, and so on. We even put a mock community. So you have 3 points. So I don't remember exactly the order, but 1 point will be Illumina, another point will be the small 263 base pair with mini ion, and the long is the 1,400 base pair with mini ion. And you can see here again that there is a good homogeneity, whatever the sequencing strategy that you use. But yeah, the thing is that since we have sequencing mistake with mini ion, it's important to have a good reference database. So it's a way also to correct a little bit of the sequencing mistake. So you have your sequence, there are some mistakes, but it fits to the nearest sequence and it gives you the right species name. I don't know if you understand myself. And it's a way to, yes, to correct a little bit that. If we don't have a good reference database, which is well completed, I think we could have some problems. And the costs, it's really cheap, 60 euros for one fungal. And the sequencing can be done in your lab. So this is really interesting methodology.